Right. Good morning, ladies and gentlemen. Thank you very much for joining our second session this morning. To those of you who are already on the call, I think we'll just give another, I would say maybe 1 minute, just to give the last attendees just a chance to join, and then we'll start very, very shortly. Right, good morning, ladies and gentlemen. I think we're pretty much just about ready to start. Guys, I just want to thank everybody, anybody who was in the first session. Thanks so much for joining the first session with us this morning. I'd also like just to thank Maria for giving the iSeq session. Maria, that was a very nice, informative session, so thank you very much for that. And then, guys, I just also wanted to let you know the session will run the same as the iSeq session did for those of you who were in that session. For those of you who weren't in the first session, what we are going to do is inside the chat window at the end of the session, we will put a participation link towards the end of the presentation just before questions and answers. That's just to allow you guys to follow that link, and then you will actually be given a certificate of attendance for actually joining and attending the session. So please stay with us till the end, and then just be sure to follow that link. Also, I'd just like to mention that we will have a question and answer session in the last sort of 10 minutes. So if you've got any questions, you're of course welcome to pose those questions in the chat window throughout the call. If it's something that we can attend to in the immediately, then we'll answer your questions. Otherwise, we'll leave the questions that you do pose in the chat window until we have our question and answer session at the end of the presentation. So, guys, I thought without any further ado, let's get started. So this session will basically focus on the next benchtop sequencing, the range of benchtop sequences by Illumina. It's known as the MiniSeq. And what we're going to do today is we're going to look at what the MiniSeq is, what its applications are, and where it sort of fits into the next-gen sequencing market of Illumina. I'd also just like to let you guys know that a lot of the content that you will see here, what we've tried to do is we've tried to mix the content up a little bit so you don't see the same content over and over. But just bear in mind, I'll mention throughout the presentation that a lot of the things you saw in Maria's presentation on the iSeq is also applicable to the MiniSeq as well as to the MySeq and the NextSeq. So the examples that we're focusing on today are by no means everything that the MiniSeq can do. It's actually a very versatile instrument. So what I'd like to just start with is just take you guys through the agenda of today's meeting. So what I'm going to start with is I'm just going to mention a couple of advantages of next-gen sequencing technology. We're not really going to go into the principles behind next-gen sequencing. I think a lot of people are already familiar with that. And for those of you who aren't, there's already links on next-gen sequencing technology on our website with other recorded webinars. So you're welcome to watch those as well in your own time. What we're then going to do is just do an introduction on the MiniSeq system, just to let you guys know where its unique points lie, why it's unique, and where its space in the next-gen sequencing market fits into. We're going to go over the sequencing overview, so just to let you guys know how the MiniSeq differs, for example, to something like the iSeq that you saw in session 1. And then look at some of the key benefits. So what are the benefits of having the MiniSeq in your lab, and where would you get the most usage out of the MiniSeq, in what sort of applications? Then after discussing those key applications, we're going to look at a couple of examples. So we're just going to look at some examples in sort of oncology workflows. We're also going to be looking at HLA typing, which Maria mentioned very briefly in the iSeq session. And we're also just going to be focusing on some general molecular biology workflows as well, followed by our question and answer session. As I said, if you've got a question, you're welcome to type it into the chat window at any time, and we can address that during the question and answer session as well. So guys, just jumping into next-gen sequencing and sort of the advantages that next-gen sequencing offers over some traditional genomic technologies. So I'm sure everybody is very familiar with qPCR, and of course, before next-gen sequencing was available, we had capillary-based Sanger sequencing as well. So we wanted to have a look at where did NGS develop, and why is NGS actually not the gold standard, but almost the way to go if you're looking for large amounts of data right now. So basically with qPCR, although qPCR is still a very routinely used assay technology in the labs, one of the drawbacks that qPCR does have, and Sanger suffers from the same issue, is that you've normally got very low complexity assays. So the issue that you've got there is you don't have the discovery power that you get with NGS, due to the fact that you are looking at fewer samples or fewer targets at a specific time, or during a specific period of time. The other issue with qPCR, and Maria mentioned this as well in session 1, was the fact that you are usually focused on single assay reactions of very small sample amounts. And usually speaking, it requires quite a cumbersome workflow. So if you guys recall the slide that Maria showed, where a qPCR run would take 11 days and you could do everything in one run on the iSeq, that basically just demonstrates the principle that the throughput of NGS and your workflows are you could get a lot more data from your workflows if you use NGS, because it's not as cumbersome and it's not as low complexity as you would get, for example, on qPCR. One of the drawbacks, for example, of Sanger sequencing is it actually has a comparatively lower sensitivity than does NGS. NGS is generally about 4 to 5 times more sensitive than Sanger sequencing. So the issue that you've got there is low sensitivity and detection power has the curious side effect that you can often miss calls, especially with DNA or RNA that is present in low copy numbers, or that obviously doesn't have, that mutation amount is quite low, or that mutations don't feature as much. You can often have missed calls, and the whole point of sequencing is so that you don't miss these calls, so that you get the best, most high fidelity data possible. The other issue that you've got with general genomic technology, so qPCR and Sanger sequencing, is that you'll see, generally speaking, your cost per sample increases linearly with the amount of samples that you're processing. So if you're processing 1,000 samples versus 10,000 samples, your cost is still going to be linearly higher. So the issue that you've got is the biggest challenge that researchers face is how do they cost effectively expand their research while decreasing their cost per sample throughput, or their cost per sample volume. And this is where NGS comes into play, is that the higher your throughput, usually speaking, the less your cost per sample are. So it's easier to expand your research as you grow. And therefore you can kind of keep the cost of the research lower. Then just another slide, I'm just going to discuss these two points at the same time, is one of the challenges that you've got with traditional methods, so for example qPCR, is that you do need a prior knowledge of a lot of the variants that you require. So for example, you need to know what gene or what target you are looking at in order to develop those or to design those primers, so that you can effectively amplify the genes of interest. With next-gen sequencing, because you've got a higher throughput, you can actually do what they call hypothesis-free mutation discovery. So because you've got such a high throughput, you don't need to necessarily focus on a couple of targets. You can, for example, on higher throughput sequences, you can do an exome sequencing or a total mRNA or even a total RNA in RNA-seq, for example. So you're getting far more data and far more depth out of a next-gen sequencing approach than you do with a traditional method. Then, as I said in a couple of slides back, Sanger sequencing generally lacks the detection sensitivity to detect low levels of DNA mutations or low copy numbers, where you don't have that issue with next-gen sequencing because you've got the slightly higher, or actually the far higher sensitivity of the technique itself. The other issue that you've got is that it's often very, very difficult to multiplex your samples, and the plexity of your multiplex analyses are generally quite low. The advantage of next-gen sequencing is you can often multiplex samples, say for example, up to 384 samples in a single sequencing run. So the plexity tends to be far higher, and that helps you with your workflows. So your workflows are not as cumbersome, and you get far more data through a single run than you would with a traditional genomic method, for example. And then, of course, just from a throughput perspective, the throughputs are much, much, much greater with next-gen sequencing than they are with traditional methods. And this is what's led to NGS being so important and so popular, for example, in things like human oncology, human genetics, immunology, and looking for the genetic drivers of disease. So let's talk a little bit about the MiniSeq system and where the MiniSeq system actually fits in. So you guys will have seen the iSeq in our previous session, for those of you who were there. The MiniSeq is basically just, if you think about it, it's basically just a larger version of the iSeq. It does use slightly different technology, but it's basically for labs that are looking for slightly higher throughputs than they can get on the iSeq. So 5 of the drivers for the MiniSeq, or 5 of the reasons why the MiniSeq is such a popular instrument, is because of its push-button simplicity. So it's basically the same as all the other Illumina sequences. It's very, very easy to set up. It's very easy to understand. It's extremely user-friendly, and it's very, very simple to define and to perform sequencing runs. The other thing with the MiniSeq is, obviously, you've still got the industry-leading quality that you find on all the Illumina sequences, so you don't have to worry about compromising quality by going for sort of a smaller system or a system on the lower throughput end. The other nice thing is the scalability. So with the MiniSeq has quite a big scalability in terms of the throughputs that you can expect from your sequencing run, and I'll discuss that in a bit more detail on the next slide. The other thing with the MiniSeq is it's also a very economical instrument like the iSeq. So for cost-sensitive laboratories that want to get started in next-gen sequencing, that require a very, very, very scalable or a very flexible system, the MiniSeq is basically one of the systems to consider, because you've got that flexibility, but it's still at a cost-sensitive price point. So it allows a lot of people to get into next-gen sequencing who would otherwise not be able to have access to the technique. And then, of course, just the flexibility in terms of the amount of different assays that can be run, or the amount of different disciplines that you can apply to your sequencing on the MiniSeq allows you to pretty much design or develop any assay or any type of routine next-gen sequencing that you would like to perform. So if you look at the scalability, basically I just decided to include this just to show you guys more or less what the throughput capability is of the MiniSeq and how it compares to the iSeq, because that gives you a bit of a better picture of how it fits into the market. So basically with the MiniSeq, you've got 2 different kits. You've got a mid-output and a high-output kit, and as you can see over here, the mid-output kit has roughly double the throughput that the iSeq 100 did. The high output kit has roughly 5 times the output that the iSeq can do. So although the sequencers can do pretty much exactly the same thing, what the MiniSeq allows you to do is it allows you to increase the complexity of your samples, or it allows you to investigate genomes, small genomes, for example, that might be slightly too large to get the correct coverage on the iSeq. So I quickly wanted just to talk about the sequencing workflow. I know Maria did already mention this in the previous session, but just sort of as a reminder to keep at the back of your mind is how the Illumina sequencing workflow is set up. So of course you do have your library preparation. So your library preparation will be anything from sample collection to sample preparation to actually using the library prep kit that you want to do to complement the investigation that you're looking for. So it might be an amplicon related assay that you'd like to run, or you might want to do a 16S metagenomics study to look at the microbiome present in your sample. You might want to do tissue typing, for example. You would select the correct library prep kit there, and then of course every system or every Illumina instrument usually has a slightly different way of performing clustering and sequencing. And then of course you've got data analysis afterwards. So the reason why I included clustering and high throughput sequencing here as sort of a blown up image of the workflow is because there's 2 sections on the MiniSeq that have basically been optimized specifically to help with the sequencing workflows and to help with actually the speed and the time that it takes to actually sequence. So 2 of the changes that Illumina made, or 2 implementations that they actually created, which are specific to the MiniSeq and actually also the NextSeq, is one thing that Illumina have done is they've looked very much at the fluidics of the systems, and on the MiniSeq they've decided to simplify the fluidics as much as possible. That has 2 side effects. You basically have quicker reagent delivery, and also you've got smaller reagent priming volumes. So it allows for your reagents to deliver quicker, and it allows for the sequencing procedure to actually occur quicker as well. And then another thing that they did do was they decided to use with the MiniSeq a 2-channel chemistry. So Maria mentioned in the iSeq that it uses a 1-channel chemistry, and she then explained how that works. On the MiniSeq, they decided to forgo the 1-channel chemistry in favor of a 2-channel chemistry instead. Now, with a 2-channel chemistry, basically what happens, which I'll show you guys now on this slide, is instead of using just a single dye, they're actually using 2 dyes, and they're taking 2 images at each sequencing cycle. Basically what happens, they use a green and a red dye. The green dye is attached directly to your thymine residues, to your T's. Your reds are attached to your C's. And then what they've done with the A's is they have attached red to half of your A's and green to the other half of your A's. And then G's are dark. So the way it works is the system will take 2 images. Clusters appearing only green will be registered as a T. Clusters appearing only red will register as a C. Clusters that will appear in both red and green will be overlaid, and they will look yellow, and those will be the A's. And images that are dark in both will be registered as a G. These are then plotted accordingly, and that is how base calling is performed. So why do the 2-channel chemistry as opposed to 1-channel chemistry? Well, there's 2 reasons for that. The first reason for using a 2-channel chemistry instead of a 1-channel chemistry is there is a slight increase in the sensitivity that you see. So by moving over to a 2-channel chemistry, you can improve the sensitivity of your assay. You can improve the sensitivity of the instrument slightly. However, by using only 2 dyes as opposed to 4 dyes, for example, if you now attach a different fluorophore to every base, you run up the costs of the reagents. So this is a happy compromise between sensitivity and cost, which is why the MiniSeq uses a 2-channel. And this just basically explains how the sequencing process is is performed. So it will incorporate the dye, take 2 images, and then the fluorophore and the terminator group will be cleaved before the next cycle carries on. And that is how base calls are performed using only a 2-channel chemistry. So by using 2-channel, you can increase the time you can decrease the time it takes to take all the images. So you're actually increasing sequencing time. You're decreasing sequencing time, my apologies, and you're also increasing the sensitivity of the assay. So some of the key benefits that we've already mentioned is obviously the push button simplicity. Everything can either be done on board. If, for example, you want to do basic analyses, that can all be done on instrument. You've also got the option to attach this directly to the cloud-based system called BaseSpace to do additional secondary and tertiary analyses. So what Illumina have tried to do is they've tried to keep the workflow as simple as possible, and that's basically across all their sequences. So you know that when you start on a sequencing run or when you start planning an assay, it's not going to be incredibly complicated, and you don't need a bioinformatician with 10 or 20 years experience. It's going to be something that you can learn and do yourself. We've already spoken about how economical, scalable, and flexible the system is. And then, of course, we've got the industry-leading data quality that Illumina is famous for. So where does it actually fit into the market? Well, it fits into any labs that have that want to adopt sequencing and that need a relatively low capital expense in order to be able to get a sequencer. So it basically just gets you out of those traditional techniques such as qPCR and more into the scope and space of next-gen sequencing. As I said, everyday users can use the system because there's simple onboard data analyses and the workflows are very, very simple and streamlined as well. And then it supports a range of targeted DNA and RNA applications due to the fact that it's extremely scalable and it's got that higher throughput than the iSeq offers you. So you've got more flexibility in terms of the library preps and the experiments that you can run on the system. So let's have a look at some of the key applications. Well, basically, the applications are very, very similar to the iSeq, and pretty much anything that can be done on the iSeq can be performed on the MiniSeq as well. However, your plexity can increase. So you can now add more samples and you can investigate more samples at the same time on the MiniSeq than you can, for example, on the iSeq. Things that are of particular interest is things like small RNA, targeted RNA, resequencing, any oncology workflows or enrichments. They can all be performed on the MiniSeq. So we'll quickly go into a couple of examples here. So we're going to give a couple of oncology workflow examples. We'll give one general diagnostic workflow and then a couple of molecular biology workflows as well, just to give you guys an idea of the flexibility and the amount of library prep kits that are available for any lab that pretty much wants to get into next-gen sequencing. There's, generally speaking, a library prep kit that's suitable for you and for your research needs. So if we look at some of the oncology workflows, if you look at, for example, a standard tumor progression pathway, you will see you basically have a normal set of cells and they will undergo some neoplastic changes, and they might be any somatic mutations, there might be gene expression or maybe even epigenetic changes or chromosomal abnormalities, and these can then result in tumor growth and tumor progression. So what you actually want to analyze in an oncology workflow is anything starting from the neoplastic change all the way to reoccurrence of cancer, should that happen. Now, there are many techniques and many options available from Illumina, but 3 that we'll focus on today is the TrueSight RNA pan-cancer, which focuses on 7 different types of cancer, including things like gastric and esophageal and lung cancer. Myeloid, which will basically look at your leukemias, so your most common types of leukemias, and then, for example, your tumor as well, your TrueSight Tumor 15, which focuses on a couple of genes that are either tumor suppressor genes or cell cycle related genes as well. So if we look at the pan-cancer gene, so basically this just gives you bit more background on the library prep kit itself. So if you look, you need very, very little DNA or RNA starting material, sorry. So generally speaking, all our library prep kits need very, very low amounts of starting RNA. And the nice thing about a lot of our onco panels is that they support FFPE as well. So these will be RNA and things gathered directly from biopsy tissue samples. Basically, the pan-cancer focuses on 1,385 genes. So you can look, it's quite a large panel of genes. Now imagine doing that on PCR would be actually quite challenging. This focuses on 1,385 genes, and a single run can accommodate up to 8 samples. So you're looking at very, very powerful amounts of data that you can get from one library prep kit on the MiniSeq. The other nice thing is you're looking at just a 4-day throughput from sample to answer. And that is much, much, much quicker than you would get with a traditional technique, for example. So it does help in early diagnosis, or it does help in emergency diagnoses as well, for example. This just gives you a bit of a feel for what the workflow of the pan-cancer looks like. So library prep would usually run over about 2 days, followed by about a 1-day sequencing, and then we've obviously got all the technologies and apps already available on BaseSpace. Some of these are also available in Local Run Manager, which will be directly on board on the instrument. So you get a complete workflow, and the workflow will be including data and report generation as well. So I also just wanted to discuss the library prep workflow itself on the pan-cancer, because we haven't actually discussed any library prep workflows. But basically, a lot of our library prep workflows that look at targeted genes use enrichment-based bead capture technology. So what would happen is you would start out with your sample material, and for example, on an RNA library prep kit, you would do standard reverse transcription PCR until you get your cDNA. After that, you would do adapter ligation and PCR amplification. But now we're only interested in a couple of genes. So what we're doing is we're using special hybridization probes that have beads on them. And what we're doing is we're actually hybridizing to our genes of interest and using these beads to actually capture only the strands that we're interested in. We then do a secondary PCR amplification, and we will then sequence only the 1,385 genes, for example, that are unique to the pan-cancer kit. So that way, you're actually being very, very, very specific in what you're sequencing, and that allows you to multiplex samples with greater efficiency and also get greater coverage for more accurate sequencing data. What you can investigate with RNA pan-cancer gives you very many clues into things like gene fusion, gene expression changes, and even RNA variants, which might contribute to a cancer disease state. Another one that we'll focus on now is the myeloid, which focuses on your 7 most common myeloid leukemias. Again, you need very, very little sample input. You're looking at only about a 7-hour preparation time with actual hands-on time of 3.5 hours. This obviously excludes your sequencing time, but your entire sample to data workflow is only about 3 days. You're looking at the 54 most common genes found in these myeloid cancers. And again, you're looking at a plexity of about 8 samples per run if you were looking at the MiniSeq specifically. This gives you, again, an idea of the throughput or of the workflow of the kit itself, as well as, again, your analyses modules, which are available directly on the instrument or available via BaseSpace as well, to give you a complete workflow. So these will be the 7 myeloid leukemias that you're looking at, and you've got a very, very, very low limit of detection in terms of the frequency of these genes. Also, greater than 95% of the amplicons from this kit, those 54 genes, will sit at 500 times coverage. And that's very, very, very important, because then you know that if you're doing variant calling, any variant, single nucleotide variants, you will be able to tell with certainty and high accuracy. Then last, we're just going to focus on the tumor, the TrueSight Tumor 15. Once again, you're looking at a 3-day analysis from sample to data. These are the 15 genes that you're analyzing. Again, it's only a 2-day period, very, very little sample input volume that's required. And you're looking again at 8 samples per run for a total of 3 runs per kit. So one thing that Maria did discuss in the first session was HLA typing or tissue typing. So I just thought I'd mention that in terms of sort of a diagnostic workflow. So this is one of our kits that actually requires or includes long-range PCR. So basically, with the loci for your HLA, which is human leukocyte antigen, your HLA markers generally are quite large. As you can see here, their links are given there. So you need to use long-range PCR in order to basically amplify that entire amplicon, and then that amplicon then obviously gets fragmented further during library preparation and sequencing. You've obviously got a completely integrated solution for data analysis and report generation using the Connexio Assign software, which will basically look through all your 11 HLA loci. I know only 8 are listed here, but you've actually got a total of 11. And this will allow a complete integrated tissue typing solution for patients that are looking for transplant viability, for example. The advantages of this kit is that you get deeper sequencing, long inserts can be performed, and you get paired-end sequencing as well, which is your forward and reverse sequencing. And all of this culminates in very accurate, very unambiguous, very high resolution HLA typing, so that you know your sample, the fidelity of your samples, and your reports are basically diagnostically viable. Again, as I said, it's a complete workflow. So Illumina is very keen on providing complete workflows so that you don't have to take your data and use third-party apps to do analyses on, and you don't require highly skilled bioinformaticians in your divisions in order to be able to use Illumina technology. So I just wanted to get to the last section, which is just a couple of molecular biology workflows on the MiniSeq. So we'll focus on sort of another 3. So one thing that's quite important is obviously small genome sequencing. So small genome sequencing, for example, viral sequencing and bacterial sequencing, they have very important applications in areas like virology and bacteriology. Also, you can map novel genomes, you can finish certain genomes, and you can also perform comparative studies between maybe divergent colonies or between divergent strains, for example. The MiniSeq, of course, allows for simultaneous sequencing of multiple samples, and because of that higher throughput, it allows you a bit more flexibility in small genome sequencing as well. So there are a couple of library prep kits which are obviously compatible with small genome sequencing. And one of the kits that you would probably use for this purpose on the MiniSeq would be the Nextera DNA Flex, which Maria discussed in session 1. So what happens if you have a library prep that you're interested in, but there isn't a custom solution, there isn't an off-the-shelf solution, rather. So then you can actually design your own one. And in this case, one of the AmpliSeq kits would be the AmpliSeq Custom DNA. And that's designed specifically for use with things like the iSeq and the MiniSeq. What that allows you to do is you might have a couple of genes that you want to investigate, and what you can then perform, or what you can then do, is develop your own custom DNA panel, which Illumina will then create for you, and then you will investigate your own amount of genes. So these can be plant genes, viral, bacterial, human. So you could do, for example, a focused immunology study, or you might want to do a structural variation study on some of the structural genes present in a specific cell type. So Illumina provide this service. So if you ever need a custom DNA panel, for example, or a custom panel, you can either contact us directly, or you can use the resources on Illumina's website to design your custom panel for you. And again, there is an available app or an available resource on BaseSpace to allow you to be able to perform these without having to worry about performing data analysis, or you now have a custom panel and now you're not too sure what to do with the data that you get, or that the data that's generated. All that you would simply do is you could upload a reference genome, for example, of the species that you're investigating, and you can perform your alignments and your variant calling directly. Right, so this just shows you again the custom DNA sample. It is compatible with FFPE as well, so you can use this again in an oncology environment if you would like to. The hands-on time is basically 3 hours plus 6 hours in terms of other sample preps and things like that. So again, you're looking at a 2 to 3 day full workflow, and you will have your answers there. The other nice thing is there's already an improved workflow, and this workflow is also automation friendly. So if there's any automation capability that you would like to do, or any automations that you would like to include, you're welcome to speak to any automation partners as well to get this kit automated on a larger scale. Again, there is 2 different kit sizes, but depending on the amount of genes that you have under investigation, you can perform sort of up to 384 samples with 384 plexity in terms of indices, and combine all of those on a single run if it's only a few genes that you're investigating. Now I'd just like to talk about small RNA. So where does small RNA fit in, and why is small RNA investigation quite important? So small RNA investigations are quite important in a number of different things. Typically, by looking at the small RNA abundance, you could look at things like transcription regulation, and it's often important in certain disease states. So, for example, it might have a lot of usage in oncology. It might have a lot of usage in immunology as well. By looking at the spread of your small RNAs and your microRNAs, you can get a slightly better picture, or you can get a more complete picture of what is actually happening inside the cell, and you can then compare that between a disease state and a normal state, for example, or between an oncology biopsy versus a normal biopsy in the same sample or in the same patient. So what you would have to do is, with the small RNAs, the reads are basically aligned against databases that already exist for mature microRNAs, small RNAs, and there's also a genomic reference that you can insert, and these will be compared against all 3 of those databases. And then what you can do is you can build an abundance of small RNA species or microRNA species to give you a better idea of the transcription regulation or gene expression status that's happening within the cell at a specific point in time. So again, it's a very small workflow. You're looking at less than 2 days, and you would basically just use the small RNA analysis module, which will be available either on BaseSpace or on Local Run Manager, to give you a complete answer and a complete workflow. Right, so the simplified workflow basically just means that you need only a single gel purification. You've got 24 samples per kit, and you're looking at a total of 1 day prep. So this is much easier than handling small RNAs or microRNAs, for example, in a PCR-based environment, and you get far more data out of the same amount of in fact, it'll take you far less time as well to get a lot more data. So I just wanted to end off with just this slide as well, just to show you guys more or less, I know Maria mentioned this as well, just to show you guys where the sequences are placed in the market or where they're spaced, and kind of more or less what the take-home message is in terms of the applications that you're going to be using them for. So you will see that the MiniSeq is placed well, it is placed very nicely in a gap between the iSeq and the MySeq in terms of the output that the system is can perform with the amount of genes or gene variants that you're experiencing or that you're investigating. So any of the kits that you're looking at, for example, with the iSeq, you could do on the MiniSeq as well. Obviously, your plexity will just be increasing. But where the MiniSeq is sort of very important is for things like immune responses, focus panels. You could do BRCA, so it gives you a lot of sample plexity on a BRCA panel. Any cancer or oncology hotspots that you're looking for. And then, of course, any germline variations or germline mutations as well. So any targeted based research that you're trying to do on a higher throughput scale or with a couple more samples, increasing your sample plexity, the MiniSeq is basically the instrument that fits well within that market space. So guys, I just would like to invite you guys just to ask a couple of questions if there's anybody that has got any questions. If you do have questions and you think about the questions later on, you're obviously welcome to either email myself or you're welcome to email our Illumina questions mailbox as well. And basically what we can then do is we can determine what your needs are and what your questions are and go from there as well. So I invite anybody for any questions that you guys might have on the MiniSeq or library prep kits or some of the workflows even.
So John, maybe just from the chat box, there's a few questions with regards to whether the slides would be available. Just to let everybody know that all the presentations for the entire series would be available for download on our website. They are quite big. If you need it sooner, like I see, there's a few participants that would like it sooner, we can share a link where it can then be downloaded from. And then Fadila would like to know, does Separations offer bioinformatics analysis or support?
That's a very good question, Fadila. Thanks so much for the question. So Separations can obviously help you with your workflows. So if you do have, for example, an off-the-shelf solution that you need secondary analysis training on or secondary analysis feedback on or something like that, yes, we can stand by you for any basic help or basic training in terms of your bioinformatic analyses. If you do require anything further than that or anything more complicated than that, then it's still possible to organize that training with you, for you with Illumina. So Illumina can then provide you with a custom training solution based on your bioinformatic requirements. I just wanted to find out if there's anybody in the chat who's actually got a MiniSeq or is working with a MiniSeq. I know we have MiniSeqs in the country. I don't know if any of our customers who are on the call are actually running on a MiniSeq currently. Right. It doesn't look like we've got any MiniSeq customers in the call right now, but I welcome any other questions that you guys might have.
Correct. You asked whether you can please have the links for the entire series as well. Definitely, that will be posted on the website in at the end of this series. Just another week to go, then all of those would be available for download from the website, the separations website. And then it's just difficult to share all the links because they are recorded by different people. So those people have to share those specific links with the different participants. And then John, just to let you know that SMU Microenviro said that they do have a MiniSeq.
Right. I wanted to find out is what if Peggy can maybe answer. I don't know if Peggy, if you know, but is there what are you guys planning on using the MiniSeq for? Do you guys have any projects in the pipeline that you guys would like to develop or look at for the MiniSeq in future?
So John, Peggy just says here we would like to use it for microbiome studies moving forward.
That's excellent. So 16S, that would be great. It's an excellent sort of assay. It's an excellent technology. It's an excellent discipline that you guys want to use on your MiniSeq. So if you guys need any further info, you guys would like to talk about any library preps or any analyses that you would like to do with regards to the microbiomes that you guys are investigating, you guys are welcome to contact us, and we'll be very, very happy to share some info with you guys there.
No further questions, John.
Great stuff. Guys, I thank you guys all for, we do have one more question that's just come in. Okay, how well does the MiniSeq handle regions of high polynucleotide repeats? Okay, that's very, very good question. Christian, to answer your question, one thing that you do, or one thing that's very important to note with a lot of the Illumina-based systems is that if you do want to look at something that has high repeats, or for example even low nucleotide variation over samples, that is possible. Usually speaking, with something like a telomeric read or very, very high or very, very long repeats, generally speaking, what you want to do there is you want to try and increase your read length as much as possible. So you want to try and span those regions to make alignments a little bit more easy. So with things like high repeats, high polynucleotide repeats, you would generally try and optimize that or opt for a MiniSeq instead. The reason why is that your MiniSeq has longer reads, so it will make alignments very, very simple for you. The MiniSeq, just like the iSeq, basically has a maximum paired end length of 150. So for an application like that, yes, you're probably better off with a MiniSeq. However, if you are looking at regions, if I expand on your question and you are looking at regions of, for example, low nucleotide variation or sort of high GC rich areas or something like that, you could always optimize that by spiking in an internal control to increase the nucleotide variance to give you a little bit better, higher quality data, if you will. Then I see we've got a couple of questions coming in here. I see Michelle asked, will you just be looking at bacteria or fungi as well in your biome? Well, that depends on you. So it depends on the genes that you're looking at. It depends on the targets that you're looking at, and it depends on whether or not you're going for an off-the-shelf or a targeted solution. If you're going for an off-the-shelf solution, it's generally 16S microbiome from bacteria only. However, you do have the ability with a custom panel, for example, to look at mycology as well. Okay, would you be using the same kit on the MiniSeq and the MySeq for microbiome studies, or are they specific for each instrument? Very good question, Fadila. No, so you would use the same kit, and then it's simply just the plexity of your samples that will differ. So say, for example, you're analyzing on a MiniSeq, you might do 16 samples, whereas on a MySeq, you might be able to go up to 48 samples or even higher. But the kits, the library preps, and usually speaking, the workflows will be identical over both instruments. Okay, then I see that Peggy asked you would like to focus on the difference in the abundance of certain bacteria related to vaccination. That's perfect. So the 16S library prep kit will be perfectly suited for you for that. And I could send you an application note just giving you a little bit more detail and more depth on the 16S workflow as well. Great, guys. So it looks like that's there's no more questions. We'll give 1 more minute just for any last-minute questions. And if there's no more last-minute questions, then I'd like to just thank you guys for all joining the session. I hope you guys found it informative, and I look forward to hearing from some of you in future.
Shelley, I think you can stop recording.